Bioluminescence transforms the deepest corners of our world into a mesmerizing, living galaxy of cold light. As we drift into this luminous world, please gently tap the like button to help this channel glow, just like the creatures we are about to explore. At its core, bioluminescence is one of the most elegant chemical reactions in the natural world, a phenomenon often referred to as cold light, because it generates illumination without generating heat. This is a critical distinction from the way humans have historically created light, which usually involves burning something or heating a filament until it glows. In the quiet darkness of the deep ocean or a damp forest, a biological creature produces light through the interaction of two specific types of molecules, a light-emitting pigment called luciferin and an enzyme called luciferase. When these two meet in the presence of oxygen, a rapid oxidation reaction occurs. The luciferin is oxidized, and in that transition, it releases energy in the form of a photon, a single packet of visible light. The names of these molecules are derived from the Latin word for lightbringer, lending a poetic weight to the chemistry. It is a process that sounds almost magical, yet it is grounded in strict physical laws, allowing organisms to carry their own stars within their bodies, independent of the sun or moon. The efficiency of this biological light is something that human engineering has struggled to replicate for centuries, as nature has perfected an energy system that is nearly 100% efficient. Consider for a moment the incandescent light bulbs that illuminated our homes for decades. Those bulbs were notoriously wasteful, converting only about 5 to 10% of their energy into visible light, while losing the rest as heat. Even our modern LED technologies, which are vastly superior, still grapple with energy loss. In contrast, the chemical reaction of bioluminescence is incredibly precise, meaning that almost every bit of energy put into the reaction comes out as light. This is why a firefly doesn't burn itself when it flashes, and why a deep-sea jelly does not boil the water around it. If these creatures produced heat alongside their light, they would likely damage their delicate tissues or waste precious calories that are needed for survival in environments where food is incredibly scarce.
This mastery of cold light represents the pinnacle of biochemical engineering, a silent, cool brilliance that illuminates the dark without warming the air or water. It is important to understand that not all things that glow in the dark are bioluminescent, and distinguishing between these phenomena reveals the unique nature of this biological power. We often group anything that shines at night into one category, but science draws clear lines between bioluminescence, fluorescence, and phosphorescence. Bioluminescence is the active production of light by the animal itself, a chemical churning within its cells. Fluorescence, on the other hand, is merely the reflection of light. A fluorescent coral or a scorpion under a blacklight is absorbing high-energy light like ultraviolet and re-emitting it at a lower energy level, usually as a neon green or blue. If you turn off the external light source, the fluorescence stops immediately. Then there is phosphorescence, which is what we see in glow-in-the-dark stars on a bedroom ceiling. This material absorbs light and releases it slowly over time. Bioluminescence is unique because it requires no external trigger. It is a deliberate internal spark of life, generated by the creature's own metabolism, making it a true source of light in the absolute void of the abyss. The existence of this living light is not a singular accident of nature, but rather a recurring masterpiece of evolution that suggests glowing is a highly advantageous trait to possess. Scientists estimate that bioluminescence has evolved independently across the tree of life, not just once or twice, but at least 40 and perhaps as many as 50 separate times. This concept, known as convergent evolution, occurs when unrelated species develop similar solutions to the same problems, much like how birds and bats both evolved wings. The fact that so many distinct lineages, from bacteria and fungi to fish, squid, and beetles, all stumbled upon the chemistry for making light suggests that the ability to glow is a powerful survival tool. It is not limited to a single ancestor. Instead, it is a tool that nature reinvents over and over again. In the deep ocean, where sunlight fades into nothingness, the pressure to evolve a personal light source is immense, leading to a diverse array of chemical recipes. Each species has its own version of luciferin and luciferase, a unique chemical signature that proves life will always find a way to illuminate the dark.
While we might imagine this biological light spanning every color of the rainbow, the vast majority of marine bioluminescence manifests in shades of blue and green. This is not a stylistic choice by the animals, but a matter of physics and the optical properties of seawater. As light travels through the ocean, water molecules absorb and scatter different wavelengths at different rates. Red light, with its longer wavelength, is absorbed very quickly, disappearing after just a few meters of depth. Blue and green light, however, have shorter wavelengths and can travel much further through the water column, allowing signals to be seen from a greater distance. Consequently, most marine eyes have evolved to be incredibly sensitive to blue and green light, and many deep-sea creatures are physically incapable of seeing red at all. By glowing in blue-green hues, these animals are broadcasting on the only channel that everyone else is watching. There are rare exceptions, of course, such as the dragonfish, which can emit a red light that functions like a secret sniper scope, allowing it to see prey without being seen. But the ocean is predominantly a symphony of blue and green. One of the most fascinating general functions of this light is a defense mechanism known as the burglar alarm effect, which turns the act of glowing into a desperate scream for help. When a small planktonic creature, like a dinoflagellate, is disturbed by a predator swimming through the water, it erupts in a flash of light. At first glance, this seems counterintuitive. Why would you light yourself up when you are trying to avoid being eaten? The logic is that the sudden flash acts as a beacon in the darkness, attracting the attention of much larger predators from further away. The hope is that the larger predator will see the light, swoop in, and eat the creature that is currently attacking the plankton. It is a biological gamble. Reveal your location to summon a monster that will eat your enemy. This is the reason why the wake of a boat or a swimmer's stroke glows at night. The microscopic life in the water is panicking, flashing their burglar alarms in response to the disturbance, creating a glittering trail of distress signals that we perceive as pure beauty. Beyond screaming for help, many creatures use bioluminescence for a sophisticated form of camouflage called counterillumination. In the open ocean, there is no place to hide. There are no rocks or caves in the vast midwater zone.
If a predator is swimming below you and looking up, your silhouette will block the faint sunlight or moonlight filtering down from the surface, making you an easy target. To combat this, many animals, such as the hatchetfish and the bobtail squid, have evolved photophores, light-producing organs, on their bellies. These creatures can meticulously adjust the intensity of the light they emit to exactly match the brightness of the downwelling light from the surface. By glowing at the perfect level, they effectively erase their own shadow. If a predator looks up, they see nothing but the continuous faint glow of the ocean surface, with no dark outline to betray the prey's presence. This is essentially an invisibility cloak made of light, a dynamic form of stealth that requires constant adjustment as clouds pass over the moon or as the animal changes depth. The human understanding of this phenomenon began long before we knew about chemical reactions, with early observations rooted in simple curiosity about glowing wood and rotting meat. In the 17th century, the famous scientist Robert Boyle, often called the father of modern chemistry, conducted experiments on what was then known as shining wood or foxfire. Boyle made a crucial discovery using his air pump. When he removed the air from a jar containing the glowing wood, the light would extinguish, and when he let the air back in, the light would return. This was a monumental realization, even if its full implications weren't understood at the time. It proved that the living light required air, specifically oxygen, to exist. This connected the gentle glow of a fungus to the more violent processes of combustion and animal respiration. It was the first step in unlocking the chemical secret that bioluminescence is an oxidation process, inextricably linked to the very breath of life that sustains the organism. While many animals produce their own light through their own genetics, a vast number of species rely on a partnership with glowing bacteria to do the work for them. This symbiotic relationship is one of the most intriguing aspects of bioluminescence. In these cases, the animal does not manufacture the luciferin or luciferase itself. Instead, it grows a specific culture of bioluminescent bacteria, such as Vibrio fischeri, inside a dedicated light organ.
The animal provides the bacteria with a safe home and a steady supply of sugar and nutrients, and in return, the bacteria pay rent in the form of continuous light. The host animal often has specialized shutters or screens of pigment that it can open and close to control the light, turning the bacterial glow on and off like a biological lamp. This partnership is so specific that when a young squid hatches, it must harvest the correct species of glowing bacteria from the surrounding seawater to colonize its light organ.
Without these microscopic tenants, the squid would remain dark and vulnerable for its entire life. Finally, when we look at the distribution of this phenomenon globally, there is a stark contrast between the land and the sea. Bioluminescence is overwhelmingly a marine phenomenon. In the dark depths of the ocean, it is the rule rather than the exception. Scientists estimate that up to 76.
The names themselves are steeped in history, derived from the Latin word lucifer, which translates to light bearer. It is a relationship of perfect specificity, much like a lock and key. A specific type of luciferase enzyme will usually only interact with its corresponding luciferin. When they meet under the right conditions, the enzyme wraps around the fuel molecule, facilitating a reaction that is one of nature's most efficient transformations of chemical potential into visible beauty. It is a common misconception that luciferin refers to a single universal compound found in every glowing creature, but nature is far more creative and diverse than that. The term actually describes a functional class of chemicals rather than a specific structure, meaning that the luciferin found in a firefly is chemically distinct from the luciferin found in a glowing mushroom or a deep-sea squid. Evolution has invented the secret of light not just once, but dozens of separate times across the tree of life, resulting in completely different chemical recipes that achieve the same radiant result. For instance, the luciferin used by fireflies is a benzothiazole derivative, a structure that requires specific cellular machinery to process. In contrast, the luciferin found in dinoflagellates, the plankton that cause sparkling waves, is a tetrapyrrole, a molecule structurally related to the chlorophyll in plants. This incredible chemical diversity suggests that the ability to glow was so advantageous that life found a way to construct it from whatever building blocks were available in different environments, creating a mosaic of light across the biological world. Regardless of the specific molecules involved, the fundamental trigger for this biological radiance is almost always the element oxygen. The chemistry of bioluminescence is in essence a very specific and controlled form of oxidation, similar in a fundamental way to how fire burns, but slowed down and managed with exquisite precision to avoid destruction. In this reaction, the luciferase enzyme helps oxygen bind to the luciferin molecule, creating a highly unstable intermediate compound, often a high-energy peroxide ring called a dioxetanone. This intermediate is in a state of extreme molecular tension, like a stretched rubber band waiting to snap. When this unstable ring inevitably collapses, it releases that stored energy.
However, instead of vibrating the surrounding molecules to create heat, the energy is released as a packet of light. The molecule effectively sheds its excess energy to return to a calm ground state, and that shedding is the glow we see. It is a moment of chemical relaxation that results in a photon traveling out into the darkness, a breath of light exhaled by the chemistry of life. In many land-based creatures, particularly the firefly, this reaction is not free. It requires a significant investment of metabolic currency to ignite the spark. Before oxygen can even enter the picture to create light, the luciferin must first be activated by a molecule called adenosine triphosphate, or ATP. ATP is the universal energy carrier of the cell, the same fuel that powers your muscle contractions and your beating heart. In the firefly's lantern, the enzyme uses magnesium and ATP to prime the luciferin, converting it into a reactive form called luciferyl adenylate. This means that every flash of a firefly is a direct conversion of the insect's biological energy reserves into light. It acts as a biological battery, ensuring that the light is only produced when the creature has the resources to sustain it. This dependency on ATP has actually made firefly luciferase an invaluable tool in modern science. Researchers use it to test for the presence of life on other planets, or to measure contamination on surfaces, because if there is a glow, there is ATP, and where there is ATP, there is life. Moving from the land to the ocean, we encounter a molecule known as sealanterrazine, which is perhaps the most popular and widespread light-emitting chemical in the marine world. Unlike fireflies, which manufacture their own fuel, many marine creatures cannot synthesize sealanterrazine themselves. Instead, they must acquire it through their diet, treating it almost like a vitamin required for illumination. This molecule travels up the food chain, creating a web of borrowed light. A small copepod might produce the chemical, which is then eaten by a shrimp, which is then consumed by a jellyfish, allowing the jellyfish to glow using the stolen chemistry of its prey. This dietary dependency explains why so many different and unrelated ocean species use the exact same type of luciferin.
Coelanterasine is an imidazopyrazonone compound, a structure that is exceptionally stable in the cold, dark depths of the ocean.
Its prevalence creates a hidden economy in the deep sea, where the currency is not just caloric energy, but the raw chemical potential to create light in the crushing darkness. There is a specific variation of this marine chemistry found in tiny crustaceans called ostracods, often referred to as sea fireflies, which use a luciferin known as vargulin. The chemistry of vargulin is remarkably stable even when the creature is no longer alive, a fact that was famously utilized during the Second World War. Japanese soldiers would collect these tiny dried crustaceans in jars, keeping them dark and dry until they needed a low-intensity light to read maps at night. By simply crushing the dried ostracods in their hands and adding a small amount of water, the rehydration would allow the dormant vargulin and luciferase to mix and react with oxygen, producing a soft blue ghost light. This light was dim enough not to be seen by enemies from a distance, yet bright enough to illuminate a piece of paper. It serves as a testament to the robust nature of these chemicals. They are stable biological tools that can wait in suspended animation for years, only releasing their stored photonic energy when the final ingredient, water, is reintroduced. While insects and crustaceans often flash on and off, bacterial bioluminescence operates on a completely different rhythm, creating a continuous, unwavering glow. The chemistry here is tied directly to the bacteria's metabolism and respiratory chain, functioning as a bypass for their normal energy production. These bacteria use a form of luciferin involved with riboflavin, specifically a molecule called reduced flavin mononucleotide, alongside a long-chain fatty aldehyde. As long as the bacteria are alive, breathing oxygen, and have a food source, the reaction runs in a constant loop, creating a steady beam of blue-green light that does not blink. This is why the light organ of an anglerfish, which is filled with these symbiotic bacteria, glows continuously rather than pulsating. The bacteria are in a state of constant chemical oxidation. This continuous glow is chemically expensive, consuming a significant portion of the distinct single-celled organism's energy, which suggests that the evolutionary benefit of being visible in the dark must outweigh the heavy metabolic tax of keeping the lights on permanently. One of the most astounding aspects of this chemical process is its efficiency, often referred to as cold light.
In the world of human engineering, generating light usually generates a byproduct of heat. An old-fashioned incandescent light bulb, for example, is incredibly wasteful, converting only about 10% of its energy into light while losing 90% as heat. If a firefly were that inefficient, the heat generated by its flashing abdomen would likely cook it from the inside out. However, the luciferin-luciferase reaction is a marvel of quantum efficiency. In many species, nearly 100% of the energy released during the chemical reaction is converted directly into light, with almost zero energy lost as thermal radiation. This extreme efficiency is achieved because the physical structure of the enzyme holds the molecules in a rigid embrace, preventing them from vibrating and dissipating energy as heat. It is a level of energetic perfection that human lighting technology, even with modern LEDs, still strives to fully replicate. While oxygen is the primary oxidizer, the trigger that actually starts the reaction can vary, and in many marine species, the key is a sudden influx of charged ions, specifically calcium. In these systems, the organism does not wait for oxygen to diffuse into the cell to start the glow. That would be too slow for a defense mechanism. Instead, they use a pre-packaged molecule called a photoprotein, such as the famous aquarin found in jellyfish. Aquarin is like a loaded gun. It consists of the luciferase enzyme with the luciferin and oxygen already bound together in a stable, frozen state, waiting for a signal. That signal is calcium. When the jellyfish is disturbed, calcium ions rush into the cell and bind to the protein, causing a slight shape change that instantly releases the pent-up energy as a flash of light. This allows for millisecond reaction times, turning the creature into a reactive strobe light that can startle predators the instant physical contact is made. The final piece of this chemical puzzle is the color of the light, which is determined not just by the fuel molecule, but by the subtle architecture of the enzyme that holds it. Interestingly, the same luciferin molecule can produce different colors, ranging from emerald green to yellow to orange, depending entirely on the structure of the luciferase enzyme hugging it.
Think of the enzyme like a pair of hands holding a glowing ball. If the hands squeeze tightly, the energy states of the molecule are altered, creating a higher energy photon like green or yellow. If the enzyme holds the molecule more loosely, or if the pH of the cellular environment changes, the light shifts toward the red end of the spectrum. This is how the railroad worm, a beetle larva, can display a red light from its head and green lights from its body, while using the exact same luciferin molecule throughout its system. It essentially tunes the frequency of the light by slightly altering the shape of the protein container, playing the electromagnetic spectrum like a musical instrument. Leaving the sunlit surface behind and descending into the midnight zone of the ocean, we encounter the iconic deep-sea anglerfish, a creature that has mastered the art of living light in eternal darkness. What makes the female anglerfish's glowing lure, or esca, so truly remarkable is not just that it glows, but that the fish itself cannot produce the light on its own. Instead, it relies on a specific species of bioluminescent bacteria that live inside the lure in a symbiotic relationship. These bacteria are not passed down from mother to daughter, which means every young anglerfish must capture its own glowing bacterial colony from the seawater as it matures, locking them inside a specialized pore that eventually closes to form the light organ. Once the bacteria are established, the anglerfish acts as a biological dimmer switch, controlling the brightness of the lure by regulating the flow of oxygenated blood to the esca, effectively pulsing the light to attract prey in the crushing blackness. In the oxygen-starved depths where the vampire squid resides, bioluminescence serves a purpose far different from the aggressive hunting of the anglerfish, functioning instead as a mesmerizing defense mechanism in a slow-motion world. The vampire squid, which is actually a unique relic that is neither a true squid nor an octopus, is covered in tiny light-producing organs called photophores that it can turn on and off at will, creating disorienting patterns of light to confuse predators. However, its most spectacular trick occurs when it is severely threatened. Rather than ejecting black ink, which would be invisible in the dark deep sea, the vampire squid expels a thick cloud of mucus containing glowing blue orbs of bioluminescence.
This glowing cloud can persist for nearly 10 minutes, creating a dazzling, luminous smoke screen that sticks to the predator and startles it, allowing the vampire squid to slip away into the shadows, while its attacker is left fighting a phantom of light. Drifting quietly through the mesopelagic zone is the cookie-cutter shark, a small but fearless predator that utilizes bioluminescence for one of the most deceptive forms of camouflage, known as counter-illumination. The entire underside of this shark is covered in light-emitting photophores that glow with a spectral match to the faint moonlight filtering down from the surface, effectively erasing the shark's silhouette against the lighter water above. However, there is a deliberate flaw in this camouflage, a small dark patch near the throat that does not glow, which acts as a lure by mimicking the silhouette of a much smaller fish to predators looking up from below. When a tuna or swordfish approaches what it thinks is a small meal, the cookie-cutter shark waits until the last second to strike, using its suction cup lids and serrated teeth to gouge a perfectly round plug of flesh from the larger animal before vanishing back into the dark. The marine hatchetfish takes the concept of counter-illumination to an architectural extreme, possessing a body that is compressed so thinly it resembles a silver coin, making it nearly invisible when viewed head-on. To handle the issue of its silhouette when viewed from below, the hatchetfish is equipped with rows of downward-facing photophores that run along its belly like the lights on an airport runway. These light organs are highly sophisticated, equipped with internal reflectors and lenses that direct the light strictly downward, and the fish can adjust the intensity of this glow to perfectly match the ever-changing brightness of the downwelling sunlight as clouds pass overhead or the sun creates an angle in the sky. This biological cloaking device renders the hatchetfish virtually two-dimensional, a phantom that exists but casts no shadow, allowing it to hide in plain sight within the twilight zone, where shadows are the primary way predators hunt. While most deep-sea creatures have evolved to see and emit blue light, which travels furthest through water, the stoplight loose-jawed dragonfish has evolved a secret weapon that functions like a sniper's night vision goggles.
This predatory fish is one of the very rare creatures capable of producing red bioluminescence, a wavelength of light that is absorbed so quickly by water that almost no other deep-sea animals have eyes adapted to see it. By projecting a beam of red light from a specialized photophore under its eye, the loosejaw can illuminate its prey without the prey ever knowing it is being watched, effectively hunting with an invisible flashlight. As we drift deeper into these dark waters and consider the quiet isolation of the loosejaw, I'm curious, do you find the idea of the deep ocean comforting in its eternal silence, or perhaps a little unsettling to think about? In the coastal waters of Japan, the firefly squid creates one of the most spectacular bioluminescent displays on Earth, gathering in the millions during spawning season to turn the surface of Toyama Bay into a glowing cobalt blue wash.
Each tiny squid, measuring only about 3 inches long, is equipped with roughly 800 tiny photophores scattered across its entire body, which can be triggered in unison or in complex rippling waves. The tips of their tentacles contain the largest and brightest photophores, which can flash powerfully to startle predators, while the smaller lights across their skin are used for communication and camouflage. During the mating season, these lights allow the squid to signal to one another in a complex visual language, creating a sea of glittering stars that mirrors the night sky above, a phenomenon so bright that local fishermen can work by the light of the catch alone. The barrel-eye fish possesses one of the most unusual adaptations for detecting bioluminescence, featuring a transparent, fluid-filled dome on top of its head that houses its sensitive barrel-shaped eyes. These tubular eyes are typically directed upward to scan for the faint silhouettes of prey against the surface light, but they are also capped with bright green lenses specifically designed to filter out sunlight and enhance the glow of bioluminescence. This filtration allows the barrel eye to spot the faint bioluminescent glow of jellies and siphonophores, creatures it often steals food from. The transparent shield protects the delicate eyes from the stinging tentacles of these jellies as the fish maneuvers carefully to snatch prey, effectively looking through its phone skull to navigate the hazardous glowing minefield of the deep ocean.
The Hawaiian bobtail squid has developed a symbiotic relationship with bioluminescent bacteria so precise that it operates like a clockwork machine, engaging in a daily cycle of acquiring and expelling its living light source. Housed within a specialized light organ in the squid's mantle, the bacteria Vibrio fischeri are fed a sugar and amino acid solution by the squid in exchange for producing light that the squid uses to match the moonlight and hide its shadow from predators on the seafloor. Each morning, as the squid buries itself in the sand to sleep, it expels about 95.
Meanwhile, the right eye is small, normal-shaped, and permanently directed downward into the abyss to scan for bioluminescent flashes from predators or prey lurking below. This evolutionary divergence allows the squid to simultaneously monitor 2 different worlds of light, the fading solar realm above and the sparking bioluminescent realm below, making it a perfectly adapted sentinel of the twilight zone. In the cool, dark waters off the Pacific coast of North America, there drifts a seemingly unassuming creature known as the crystal jelly, or Aquaria victoria, which holds within its delicate, transparent body a secret that revolutionized modern science. While many jellyfish produce a brief flash when disturbed, the crystal jelly produces a steady, glowing ring of green light around the rim of its bell, a phenomenon that puzzled researchers for decades because the chemical reaction inside the jellyfish actually produces blue light, not green. This discrepancy led to the discovery of a remarkable protein called green fluorescent protein, or GFP, which absorbs the blue light and re-emits it as a brilliant green glow, functioning much like a natural transformer of energy. This discovery was far more than a biological curiosity. Because GFP can be attached to other proteins without harming living cells, it became a microscopic torch for scientists, allowing them to tag and watch biological processes happen in real time, from the spread of cancer cells to the development of nerve connections in the brain. It is a humbling thought that much of our current understanding of the inner workings of human health illuminates our laboratories only because of a simple, drifting jelly that has been pulsing in the silent tides for millions of years. Drifting deeper into the midnight zone, where sunlight never reaches, we find the Atolla jellyfish, a deep red creature that employs one of the most psychologically complex defense strategies in the animal kingdom, often referred to as the burglar alarm display. Most deep-sea creatures use bioluminescence for camouflage or to attract a mate, but when the Atolla is attacked by a predator, it does not try to hide. Instead, it erupts into a frenzied series of expanding blue flashes that pinwheel around its body like the lights of a police car.
This dazzling light show is not meant to frighten the attacker, but rather to scream into the darkness, illuminating the immediate area to attract a much larger predator, one big enough to eat the creature that is currently attacking the jellyfish. It is a high-stakes gamble of the enemy of my enemy is my friend, weaponizing the fear of being seen in the eternal dark, and ensuring that any creature daring enough to take a bite of the Atolla risks becoming a meal itself in the sudden, exposing glow. While true jellyfish pulse with chemical light, the mesmerizing comb jellies, or ctenophores, are often mistaken for being bioluminescent due to a completely different optical trick of physics called diffraction.
If you have ever seen footage of these ancient oval-shaped wanderers, you likely noticed rows of beating cilia running down their sides, shimmering with a rainbow of colors like a living prism. But this iridescence is merely the scattering of ambient light, not the production of it. However, many comb jellies do possess true bioluminescence, which is almost always a ghostly blue-green, stored in canals that run beneath their comb rows, allowing them to trace their own geometric shapes in the absolute darkness. Unlike other marine invertebrates that flash only when touched, some comb jellies can release a glowing secretion into the water when threatened, leaving behind a sparkling phantom of themselves to distract predators, while the jelly itself slips silently away into the abyss. Among the drifting relatives of the jellyfish are the siphonophores, complex colonial animals that can stretch for meters in the deep, and one genus in particular, Irena, has evolved a lure that defies the usual rules of marine light. While almost all bioluminescence in the ocean is blue or green, because those wavelengths travel furthest through seawater, the Irena siphonophore produces a rare red light, generated by specialized stalks that twitch and flick to mimic the movement of small copepods. In the deep ocean, most creatures have lost the ability to see red light, since it is filtered out by the water column so quickly. But the fish that Irena preys upon mistake these glowing red lures for the silhouette of a small shrimp or worm.
By utilizing a color that is nearly invisible to the wider ecosystem, this siphonophore creates a private trap, a secret signal that only attracts its specific prey, dangling a glowing red bait in a world that is otherwise entirely monochrome. The most commonly witnessed form of marine bioluminescence, the kind that paints crashing waves with neon blue or leaves glowing footprints in the sand, is caused by microscopic plankton known as dinoflagellates, specifically a species often called sea sparkle. These tiny, single-celled organisms drift by the billions in surface waters, and their light is triggered by mechanical stress, the physical deformation of their cell walls caused by the motion of waves, the stroke of a swimmer's hand, or the wake of a boat. The chemical reaction inside them is instantaneous, taking only 20 milliseconds to reach peak brightness, which is faster than the transmission of a nerve impulse in a human, creating a defense mechanism designed to startle predators. Have you ever walked along a beach at night and seen the water glow, or perhaps wished you could see such a thing? It is a reminder that the ocean is not just water, but a living soup of light, waiting for the slightest touch to wake up. These glowing dinoflagellates possess a sophisticated internal clock, a circadian rhythm that strictly controls their ability to produce light, ensuring they never waste energy glowing during the day when the sun would wash them out. Even if you were to take a jar of water filled with these organisms and place it in a dark closet at noon, shaking the jar would result in darkness. The plankton know it is daytime and chemically disassemble their light-producing luciferin to conserve resources. As evening approaches, they synthesize the necessary chemicals again, recharging their biological batteries in perfect sync with the setting sun, preparing to turn the ocean surface into a minefield of light. This cycle persists even in the laboratory under constant dim light for several days, driven by an ancient genetic timer that aligns their tiny lives with the rotation of the Earth itself. Descending to the realm of crustaceans, we find the ostracods, often called sea fireflies or seed shrimp, which are responsible for some of the most romantic and precise light displays in the Caribbean coral reefs.
Unlike the reactive flashes of plankton, male ostracods perform complex, voluntary mating dances, swimming upward from the seafloor while releasing pulses of blue light to create vertical pillars of glowing dots in the water column. Each species has a specific code. Some produce short, rapid staccato blinks, while others release long, languid trails, ensuring that females, waiting in the darkness below, swim up only to the display that matches their own species. This creates a silent underwater fireworks show that is completely invisible to the human eye from the surface, a secret courtship ritual occurring in the gaps between the coral, driven by the desire to be seen in the dark. The light of the ostracod is so chemically stable and potent that during the Second World War, Japanese officers famously used dried ostracods, which they called umihotaru, as a makeshift low-intensity light source for reading maps in the trenches at night. The dried crustaceans, when crushed in the palm of a hand and moistened with just a tiny bit of water or saliva, would reactivate their dormant chemicals and emit a soft, cool blue glow. This light was bright enough to illuminate the lines on a map, but dim enough to avoid being spotted by enemy planes or sentries from a distance, effectively turning the biology of a tiny sea creature into a piece of tactical military equipment. It is a testament to the resilience of the luciferin-luciferase reaction that the potential for light can lie dormant in a dried shell for months, waiting only for hydration to unlock its energy once more. Another master of defensive light is the copepod, a tiny teardrop-shaped crustacean that serves as a primary food source for countless marine animals, yet has developed a depth charge strategy to escape death. When a copepod detects the pressure wave of an approaching fish, it does not just flash, it violently expels a cloud of sticky, glowing slime from glands on its body, creating a bright, lingering phantom in the water. This glowing cloud acts as a decoy, hovering in the water and baffling the predator, which often bites at the light, while the copepod itself uses the distraction to dart away into the surrounding shadows. This is the underwater equivalent of a smoke bomb, but constructed entirely of cold fire, allowing one of the ocean's smallest creatures to outwit hunters hundreds of times its size by simply leaving a ghost of itself behind.
Finally, we look to the Antarctic krill, billions of which swarm in the cold southern oceans, possessing highly advanced photophores that function almost like flashlights, complete with lenses to focus the light and muscles to aim it. These shrimp-like creatures use their lights primarily for counter-illumination, a form of camouflage where they emit light from their undersides to match the intensity and color of the moonlight or starlight coming from the surface above. By perfectly matching the downwelling light, the krill erase their own silhouettes, making them invisible to predators looking up from below, effectively disappearing by glowing. They can even tilt their photophores to account for the angle of the light as they swim, a physiological feat of engineering that turns their entire bodies into active cloaking devices, hiding them in plain sight within the luminous blue of the polar seas. Leaving the mysterious depths of the ocean where light is a tool for survival in the abyss, we find that the phenomenon of living light is not exclusive to the water, but paints the terrestrial night with equal brilliance, particularly within the enchanting world of the firefly. Among the thousands of firefly species, there exists a rare and spectacular phenomenon known as simultaneous bioluminescence, primarily displayed by the species Photinus carolinus, found in the Great Smoky Mountains. For a few weeks each year, usually in late May or June, the male fireflies in these dense forests synchronize their flashing patterns in a way that defies the chaotic randomness we usually associate with insect swarms. Instead of twinkling independently, thousands of beetles illuminate the dark forest canopy in a perfect, pulsating rhythm, flashing 6 to 8 times in quick succession, followed by a specific period of total darkness. This synchronization is a behavior known in physics as coupled oscillation, where each individual adjusts its timing based on the flashes of its neighbors, creating a rolling wave of light that allows females on the ground to distinguish their own species from the visual noise of the forest, a mesmerizing light show that turns the woods into a breathing, living entity of light. While the beauty of these flashes captures our imagination, the physics behind the firefly's lantern is a marvel of energy efficiency that human engineering has struggled to replicate for decades.
The light produced by a firefly is often referred to by scientists as cold light because it generates almost no thermal radiation during the reaction. In a standard incandescent light bulb, roughly 90% of the energy is wasted as heat, with only 10% emitted as visible light. And even modern LED technology, while vastly superior, still grapples with heat dissipation. The firefly, however, operates at nearly 100% quantum efficiency, meaning that for every single photon of light emitted, virtually no energy is lost as waste heat. This is crucial for the insect's survival, as any significant heat production within their tiny abdomens would be fatal, cooking them from the inside out. The precise chemical reaction involves the oxidation of luciferin catalyzed by the enzyme luciferase, a process so refined by evolution that it produces a pure, steady glow without the thermal cost that usually accompanies the generation of light in the physical universe. However, the world of the firefly is not purely a romantic dance of signals and efficiency. It also harbors a dark and deceptive secret involving the photurus firefly, often called the femme fatale of the insect world. While most fireflies use their specific flash patterns to attract mates of their own species, the female photurus has evolved the ability to mimic the flash responses of females from other firefly genera, particularly the smaller Photinus. When a male Photinus flashes his courtship signal, the predatory Photurus female replies with the exact flash pattern he is expecting from a potential mate of his own kind, luring him in through the darkness. When the unsuspecting male lands, expecting to find a partner, he is instead devoured by the photurus, who does this not just for nutrition, but to steal defensive chemicals called luciferins. The photurus cannot manufacture these toxins herself, so by eating the males of other species who do possess them, she sequesters the poison into her own body to protect herself and her eggs from spiders and birds, a chilling example of aggressive mimicry fueled by bioluminescence. The engine driving this biological light, particularly in insects, relies heavily on a molecule that is the universal energy currency of all known life, adenosine triphosphate, or ATP.
The reaction that causes the firefly's abdomen to glow is strictly dependent on the presence of ATP to activate the luciferin before it can react with oxygen. This dependency is so reliable that scientists have harnessed firefly luciferase in laboratory settings to detect the presence of life or contamination on surfaces. By swabbing a surface and adding the firefly enzyme, researchers can instantly measure the amount of light produced. Since the enzyme only glows in the presence of ATP, and ATP is only found in living cells, the intensity of the glow reveals the level of bacterial contamination. This means the same mechanism that helps a firefly find a mate in a summer meadow is used by food safety inspectors and space agencies to test for microscopic life, bridging the gap between a summer evening's wonder and advanced biotechnology. Beyond the familiar firefly, the terrestrial night is patrolled by the Phengodidae family, commonly known as railroad worms, which display one of the most unique and tactical uses of bioluminescence in the animal kingdom. These beetle larvae, which look somewhat like caterpillars, possess two different colors of light on a single body, a rare trait given that most bioluminescent organisms emit only one part of the spectrum. The railroad worm earns its name from the rows of greenish-yellow glowing spots along its body that resemble the illuminated windows of a train car at night. But its true superpower lies in the vivid red light it emits from its head. Most insects and potential prey in the railroad worm's environment are red blind, meaning they cannot see red light at all. They perceive it as darkness. This allows the railroad worm to use its red headlamp like a sniper's night vision scope to scan the environment and stalk prey, without the prey ever knowing it is being illuminated, giving the predator a stealth advantage that is almost unfair in its evolutionary perfection. While the railroad worm creeps along the ground, the tropical night air is often pierced by the erratic flight and intense glow of the click beetle, specifically those of the genus Pyralphorus, sometimes called cucabanos. Unlike fireflies, which typically house their light organs in the soft tissue of the abdomen, the click beetle boasts two distinct glowing spots on its hard thorax that look exactly like the headlights of a tiny car, in addition to a third light organ hidden under its wings that is only visible during flight.
These beetles are named for the complex spine and notch mechanism on their chest that allows them to snap their bodies with a loud clicking sound to launch themselves into the air if they are overturned. Their bioluminescence is remarkably bright and constant, often glowing continuously rather than pulsing, and is intense enough that early travelers in tropical regions were known to tie these beetles to their toes to light their path through the jungle, or keep them in small wicker cages to use as reading lamps, utilizing the insect's natural brilliance as a primitive flashlight. The terminology we use can be deceptive, for the creature often called a glowworm is rarely a worm at all, but is frequently the larval stage or the wingless female of a beetle species. This distinction highlights an evolutionary timeline where bioluminescence likely appeared in the larvae long before it was used for adult courtship. Scientists believe that the original purpose of the glow in these terrestrial insects was aposematism, a warning signal to predators indicating toxicity. Because many firefly larvae contain bitter or poisonous steroids, the glow serves as a bright, memorable do not eat sign to nocturnal hunters like toads and mice. Over millions of years, this larval warning system was retained into adulthood and eventually co-opted and refined into the complex mating signals we see today, meaning the romantic flickering of a summer night actually began as a defensive shield to keep soft-bodied larvae safe on the dark forest floor. This chemical defense is physically demonstrated through a phenomenon known as reflex bleeding, which connects the insect's internal biology directly to its light. When a firefly is threatened or handled roughly, it can voluntarily rupture membranes in its exoskeleton to bleed droplets of hemolymph, the insect equivalent of blood. This blood is loaded with the luciferins and other defensive chemicals that taste intensely bitter and can numb the mouth of a predator. The bioluminescence acts as the advertising billboard for this chemical weaponry. Predators learn to associate the specific wavelength of yellow-green light with the terrible taste and toxic effects of the hemolymph. It is a perfect communicative loop, where the light ensures the predator recognizes the prey before attacking, sparing the firefly from injury and the predator from a sickening meal, creating a truce negotiated through photons in the dark.
Traveling to the caves of New Zealand and Australia, we encounter a terrestrial bioluminescent creature that is neither a beetle nor a worm, but the larva of a fungus gnat known as Arachnocampa luminosa. These insects create spectacular galaxies of blue-green light on the ceilings of damp caves and sheltered grottos, a sight that draws tourists from around the world. Unlike the firefly's mobile hunt for a mate, the glowworm's light is a stationary trap. The larva constructs a snare of silk threads studded with droplets of mucus that hang down like beaded curtains. The larva sits in the center of this web and glows intensely, utilizing the fact that many nocturnal flying insects navigate by the light of the moon and stars. By mimicking a starry sky in the pitch-black environment of a cave, the glowworm disorients moths and small flies, luring them upward until they become entangled in the sticky silk lines, at which point the larva senses the vibration and hauls up the line to consume its prey. The complexity of these signals in the open air reaches a peak in the specific flash codes used by different firefly species to ensure they do not mate with the wrong kind, essentially speaking different languages of light. Each species has a unique Morse code, defined not just by the color, but by the duration of the flash, the number of pulses in a phrase, and the interval of silence between them. Some species fly in a specific shape while flashing, such as the J-hook pattern, where the male flies horizontally, dips down, and then flashes as he swoops back up, creating a literal letter J written in light. Others produce a long, continuous streak, or a double flicker that must be timed to the millisecond. The females on the ground are genetically programmed to respond only to the precise timing and movement of their own species, ensuring that in a meadow filled with thousands of blinking lights, the message always reaches the intended recipient without confusion. As we drift through these illuminated forests, I wonder if you have ever been lucky enough to see a firefly or a glowworm in person, and how that moment of tiny, fleeting magic made you feel in the quiet of the night. Deep in the quiet, shadowed undergrowth of ancient forests, a phenomenon known as foxfire has captivated human imagination for millennia, where decaying wood emits a soft, ghostly green light caused by bioluminescent fungi.
This ethereal glow is not magic, but the result of a complex chemical reaction within the fungal threads, or mycelium, permeating rotting logs. The primary species responsible for this haunting radiance is the honey mushroom, specifically the genus Armillaria. What makes this truly fascinating is the biological purpose behind the energy expenditure. Unlike many marine creatures that flash to startle predators, these mushrooms emit a continuous, steady light to attract insects. In the stillness of the night, beetles and flies are drawn to the verdant shimmer, landing on the fruiting bodies and picking up spores, which they then carry to new locations, ensuring the survival of the fungus. The chemistry involves a specific molecule called hispidin, an antioxidant that protects the fungus from toxic byproducts during the day, but at night is converted into a light-emitting substance, creating a rhythm of light synchronized with the setting sun. While the honey mushroom is widespread, the bioluminescent abilities of the bitter oyster mushroom, scientifically known as Panellus stypticus, reveal a curious evolutionary puzzle rooted in geography. This small, fan-shaped fungus grows on hardwoods, but if you were to walk through a forest in the Pacific Northwest of North America, you would find them completely dark, whereas the exact same species growing in the hardwood forests of the eastern United States glows with impressive intensity. This geographical disparity has allowed geneticists to study them by crossbreeding the glowing eastern strains with the non-glowing western ones, revealing that the capability to produce light is a dominant trait. The non-glowing western variety actually possesses the gene cluster for bioluminescence, but lacks the specific promoter region to turn it on, suggesting that at some point in evolutionary history, the western population lost the need to glow, perhaps due to a lack of specific nocturnal insects in that region to aid in spore dispersal, or simply to conserve metabolic energy in a different climate. Traveling across the ocean to the tiny Hachijo Island in Japan, one can witness the breathtaking spectacle of Mycena lux coli, a name that translates poetically to heavenly light mushrooms. During the rainy season, these dime-sized fungi erupt from the bark of chinkapin trees, glowing with such intensity that they can reportedly illuminate the ground around them clearly enough to see your own hands.
Unlike the bitter oyster mushroom, where only the gills or mycelium might glow, the entire surface of the heavenly light mushroom radiates a deep, mesmerizing emerald hue. The chemical reaction in these delicate organisms is so efficient that nearly 100% of the energy involved is converted into light, with almost no heat generated, a feat of cold fire that modern engineering still struggles to replicate perfectly. Walking through these forests after a rainfall feels like stepping into a dreamscape, where hundreds of tiny, living lamps transform the dark, rotting logs into constellations of green starlight on the forest floor. In the southern hemisphere, particularly in the forests of Australia and Tasmania, the concept of a toadstool takes on a spectral reality with the ghost fungus, Omphallotus nidiformis. By day, this fungus appears deceptively ordinary, resembling the edible oyster mushroom with large, creamy white, funnel-shaped caps often found at the base of eucalyptus trees. However, as twilight fades into total darkness, the white cap disappears and is replaced by a vivid, pale green luminescence that outlines the gills and margins of the fungus. This species provides a perfect example of convergent evolution and a dangerous trap for the unwary forager. While it looks like a delicious meal during the day, it is toxic, causing severe illness. Its glow is an advertisement not for humans, but for large flies and beetles, effectively using its light as a beacon to ensure its reproductive future, while simultaneously warning mammals that the glowing spectral form is not something to be eaten. Descending from the visible world of mushrooms to the microscopic realm, one of the most historical and touching interactions between humans and bioluminescence occurred during the American Civil War in a phenomenon soldiers called angel's glow. After the Battle of Shiloh in 1862, wounded soldiers lay in the mud and rain for 2 days awaiting rescue, and during the cold nights, many noticed their wounds beginning to glow with a faint blue-green light. Surprisingly, the soldiers whose wounds glowed seemed to have higher survival rates, and their injuries healed more cleanly than those without the light. We now know this was caused by Photorhabdus luminescens, a bioluminescent soil bacterium. These bacteria live inside microscopic nematodes that hunt insect larvae. When the nematode enters a host, it regurgitates the glowing bacteria, which release chemicals to kill the insect and antibiotics to eliminate competing microbes. In the cool, damp conditions of the battlefield, the soldiers' hypothermia allowed the bacteria to survive in their wounds, where the angel's glow essentially sanitized the injuries, acting as a natural, life-saving antibiotic long before the discovery of penicillin. While Photorhabdus works on a microscopic scale, other bioluminescent bacteria create phenomena so vast they can be seen from space, known as the Milky Seas effect. This rare maritime event, often recorded in the logs of 19th century sailors as oceans turned to milk or clouds, is caused by a massive bloom of bioluminescent bacteria, such as Vibrio harvii, colonizing the surface of the water. Unlike the brief, spark-like flashes of plankton disturbed by a ship's wake, a Milky Sea is a continuous, steady glow that can extend for thousands of square kilometers, persisting for days. The bacteria use a sophisticated communication method called quorum sensing. Each individual bacterium is too small to be seen, but they release a chemical signal, and only when the population density reaches a critical mass does the entire colony switch on their lights simultaneously. This creates a surreal, glowing skin on the ocean that tricks satellite sensors into thinking they are seeing clouds or snow, a testament to the power of microscopic cooperation. Returning to solid ground, the Sierra Nevada mountains of California conceal a creature that uses light as a formidable chemical warning, the Motixia millipede. These blind, vegetarian arthropods are the only known millipedes to display bioluminescence, glowing with a continuous bluish-white light as they crawl through the leaf litter. While a glow usually attracts attention, for the Motixia it serves as a stark warning sign of toxicity, a mechanism known as aposemicism. The body of the millipede produces cyanide, a lethal poison, and the intensity of the millipede's glow directly correlates to the amount of cyanide it holds. The brighter the light, the more poisonous the buck. Evolution has honed this signal so effectively that nocturnal predators like rodents, which would normally rely on scent or sound, have learned to avoid the glowing cues entirely, treating the beautiful light as a strict do not touch sign in the darkness of the mountain underbrush. In the tropical gardens and rainforests of Southeast Asia, specifically Singapore and Malaysia, lives the only known land snail capable of producing its own light, Quantula striata. Most bioluminescent animals on land, like fireflies, flash their lights to find mates, but the hiker snail operates differently. Its eggs glow continuously in the dark, turning the soil into a dim galaxy of hidden potential. As the snails hatch and mature, the ability to glow shifts to an organ near the mouth, which flashes intermittently while the snail is moving. The purpose of this flashing is still a subject of scientific debate. Some researchers believe the pulsing light helps the snails coordinate with one another in the dense vegetation, while others suggest it might be a way to startle predators, or perhaps even lure curious prey, making this unassuming mollusk a unique anomaly in the gastropod world where light is almost never a part of the anatomy. Deep within the soil of the American South, particularly in states like Georgia, lives a creature that brings bioluminescence to the humble earthworm, the Diplocardia longa. These are not your average garden worms. They can grow remarkably large, up to nearly 2 feet in length, and they possess a defense mechanism that is both sticky and radiant. When these worms are disturbed or threatened by a predator like a mole or a centipede, they exude a thick, slimy coelomic fluid that bursts into a bright bluish glow. This glowing slime is incredibly sticky, adhering to the predator and effectively tagging it with light, which makes the attacker vulnerable to its own predators while simultaneously startling it enough to let the worm escape back into the earth. The biochemistry of this worm is distinct from marine life and fireflies, utilizing a unique luciferin known as N-isovaleryl 3-aminopropanol, proving that nature has reinvented the chemistry of light multiple times in completely different environments. Rounding out the terrestrial and freshwater curiosities is the New Zealand freshwater limpet, Latia narratoides, which holds the title of being one of the very few freshwater organisms on the entire planet to exhibit bioluminescence.
While the oceans are teeming with glowing creatures, freshwater lakes and streams are surprisingly dark, ecologically speaking, making Latia an evolutionary eccentricity. Clinging to rocks in fast-flowing streams, this small mollusk defends itself by secreting a luminous green mucus when disturbed. If a trout or an eel attempts to nip at the limpet, the release of this glowing cloud serves to confuse the predator, momentarily blinding it or creating a glowing decoy in the turbulent water while the limpet clamps down or drifts away. The mechanism relies on a purple protein and a clear enzyme that mix to produce the light, a primitive but effective alarm system that has allowed this solitary species to survive in the pitch-black currents of New Zealand's rivers. Deep within the mesopelagic zone, often called the twilight zone, survival depends heavily on the mastery of light, particularly through a strategy known as counter-illumination, which is one of the most sophisticated forms of camouflage in the natural world. In this realm, the faint sunlight filtering down from the surface creates a deadly silhouette for any creature swimming above a predator looking upward, effectively turning them into a clearly defined shadow against the lighter blue background. To combat this, the deep-sea hatchetfish has evolved a narrow blade-like body lined with rows of photophores along its belly that act like biological dimmer switches. These light-producing organs emit a soft blue glow that precisely matches the intensity and color of the sunlight filtering down from above, effectively erasing the fish's shadow and rendering it invisible to predators lurking below. This is not a static process. The hatchetfish constantly adjusts the brightness of its bioluminescence in real time to match the changing light conditions as clouds pass overhead or as it swims to different depths. This active masking capability suggests a high level of neurological control, allowing the fish to disappear in plain sight by blending perfectly with the downwelling light, a mechanism that military engineers have long studied in the hopes of creating aircraft that are invisible to the eye. While the hatchetfish uses light to hide, tiny marine plankton known as dinoflagellates use it for a dramatically different purpose known as the burglar alarm effect.
These microscopic organisms drift in vast numbers near the ocean's surface, and when disturbed by the movement of a predator like a grazing copepod, they erupt in a brilliant flash of blue light. At first glance, lighting up when you are about to be eaten seems counterintuitive, as it reveals your location. But the strategy is designed to attract a secondary predator, a larger fish or crustacean that preys on the copepod.
By illuminating the immediate area, the dinoflagellate effectively paints a target on its attacker, summoning a larger hunter to eliminate the threat. This complex trophic interaction relies on the fact that light travels efficiently through clear water, signaling the presence of a meal to predators from a significant distance. Experiments have shown that when bioluminescent dinoflagellates are present, the predation rate on grazers like copepods increases significantly, while the plankton themselves are spared. It is a biological scream for help, a chemical siren that turns the dark ocean into a dangerous arena where the hunter instantly becomes the hunted, proving that in the ocean, the enemy of my enemy is my friend, provided I can signal them with a flash of light. Moving from defense to the offensive capabilities of light, the deep-sea dragonfish, specifically the species Malacosteus niger, possesses one of the most unique predatory advantages in the animal kingdom, the ability to see and emit red light. In the deep ocean, water absorbs red light wavelengths very quickly, meaning most deep-sea creatures have evolved eyes that are sensitive only to blue and green light, rendering them completely blind to the red end of the spectrum. The dragonfish exploits this by using a suborbital photophore located beneath its eye to project a beam of red bioluminescence, functioning exactly like a sniper using invisible infrared night vision goggles. It can sweep this beam of red light across the darkness to illuminate prey without them ever knowing they are being watched, as the prey physically cannot see the light hitting them. To perceive this red light itself, the dragonfish requires a special retinal pigment, a derivative of chlorophyll likely obtained from its diet of copepods, which allows it to translate the red wavelengths into a visual signal.
This private channel of communication and hunting allows the dragonfish to stalk its victims in absolute stealth, closing in for the kill while the prey remains comfortably oblivious in what it believes to be total darkness. In the oxygen-starved depths where the vampire squid resides, traditional defense mechanisms like the ink clouds used by shallow water squid would be ineffective, as black ink is useless in the crushing blackness of the abyss. Instead of ink, the vampire squid, which is actually a unique relic species bridging the gap between squids and octopuses, ejects a thick bioluminescent mucus containing thousands of glowing orbs of blue light from the tips of its arms. When threatened, it throws this glowing cloud over its head while simultaneously somersaulting away into the darkness, a maneuver that creates a dazzling and confusing display of light that lingers in the water for nearly 10 minutes. The predator is left disoriented, attacking the glowing phantom cloud while the squid makes its silent escape. This secretion is metabolically costly to produce, so the vampire squid uses it only as a last resort, preferring first to pull its webbed arms over its body in a pineapple posture that exposes defensive spines. The glowing mucus is sticky and adhesive, clinging to the predator's face or sensors, which may further irritate or confuse the attacker, providing a multi-sensory distraction that ensures the survival of this ancient, slow-moving cephalopod in a world where speed is not always an option. One of the most remarkable discoveries in recent years involves a group of segmented marine worms known as the green bomber worms, or Swimma bommi-viridis, which live near the seafloor and have evolved a sacrificial strategy involving detachable body parts. These worms possess small, fluid-filled sacs or bombs located near their heads, which are actually modified gills that have lost their respiratory function to become specialized bioluminescent grenades. When the worm is disturbed by a predator, it can release one or more of these bombs, which burst into a brilliant green phosphorescence upon detachment, glowing intensely for several seconds. The sudden flash is intended to startle the predator and draw its attention to the glowing decoy, allowing the worm to swim away in the dark. This behavior is similar to lizards dropping their tails, but with the added element of a blinding light show.
The light is produced by a specific chemistry that only activates when the bomb is jettisoned, ensuring the worm doesn't accidentally light up while the sacs are still attached. These worms can regenerate the bombs over time, ensuring that they always have a stockpile of ammunition ready for the next encounter, showcasing a remarkable evolutionary investment in distraction as a primary means of defense. Deception runs deep in the ocean, and the cookie-cutter shark employs a particularly devious form of bioluminescent mimicry to feed on animals much larger than itself. This small, cigar-sized shark spends its days in the deep and migrates upward at night, where it uses a specialized collar of dark, non-glowing skin around its throat to interrupt the green bioluminescent glow that covers the rest of its underside. While its glowing belly uses counter-illumination to hide it from below, the dark collar acts as a lure by mimicking the silhouette of a much smaller fish when viewed from beneath against the moonlight. Large predators like tuna, marlin, or dolphins see this small, non-threatening shadow and rush upward to eat what they think is a small meal,
only to find themselves face to face with the razor-sharp circular teeth of the cookie-cutter shark. The shark seizes the opportunity to latch on to the larger predator, spinning its body to carve out a perfect plug of flesh before releasing its grip. This strategy, known as aggressive mimicry, allows a relatively weak swimmer to bring the food to itself, using the allure of a full silhouette to turn a top predator into a victim of a parasitic strike. On land, the complexity of bioluminescent communication reaches its peak with fireflies, specifically in the interaction between the genera Photinus and Photurus, where light signals become a language of love and death. Male fireflies of the Photinus genus fly through the air flashing specific rhythmic patterns to signal their interest to females waiting in the grass, who respond with a precise time-delayed flash to confirm their species and readiness to mate. However, the larger female fireflies of the Photurus genus have hacked this code. They are capable of mimicking the specific flash response of the Photinus females with terrifying accuracy. When a hopeful male Photinus sees the flash, he descends thinking he has found a mate, but upon landing, he is promptly seized and devoured by the Photurus female.
This behavior, often called the femme fatale effect, allows the predatory female to obtain valuable nutrients and defensive toxins called lucibophagens from the male, which she cannot produce herself, and which protect her from spiders and birds. It is a stunning example of aggressive mimicry where the signal for reproduction is hijacked for predation, turning the very mechanism of species survival into a fatal trap. Perhaps the most iconic example of bioluminescent predation is found in the deep-sea anglerfish, specifically the females, who sport a modified dorsal fin spine called an elysium that extends over their heads like a fishing pole. At the tip of this spine sits the esca, a bulbous lure that glows with a ghostly light not produced by the fish itself, but by a colony of symbiotic bioluminescent bacteria living inside the organ. These bacteria are acquired from the seawater as the anglerfish matures and are housed in the esca, which is connected to the fish's bloodstream, allowing the host to control the light by regulating the flow of oxygen and nutrients to the bacteria. In the perpetual darkness, the glowing lure attracts curious fish and crustaceans looking for a meal, drawing them closer to the anglerfish's massive, upturned mouth, lined with translucent, needle-like teeth. The anglerfish can twitch and wiggle the lure to simulate the movement of a small shrimp, enhancing the deception. This relationship is obligate, meaning the bacteria cannot reproduce outside the host, and the anglerfish relies entirely on the light to feed in an environment where food is scarce, representing a perfect evolutionary partnership forged in the dark. Bioluminescence can also serve as a warning signal, a phenomenon known as aposematicism, which is clearly demonstrated by certain species of colonial siphonophores, gelatinous relatives of jellyfish. These drifting colonies, which can reach lengths of several meters, possess stinging tentacles packed with potent neurotoxins capable of paralyzing small fish and injuring larger creatures. To avoid unnecessary conflict that might damage their fragile bodies, many deep-sea siphonophores display bright, rhythmic flashes of blue or green light along their stems when disturbed. This light serves as a clear do not touch sign to potential predators, communicating that the creature is dangerous and unpalatable. It is the deep-sea equivalent of the bright colors seen on poison dart frogs in the rainforest. By advertising their toxicity or dangerous stinging capability through light, they prevent predators from attacking in the first place, saving energy that would otherwise be spent on regeneration or active defense. Research suggests that visual predators in the deep sea have learned to associate these specific light patterns with pain, effectively creating a learned avoidance behavior that protects the slow-moving siphonophores as they drift through the currents. Among the bottom dwellers, the scale worm utilizes a brilliant distraction technique involving detachable, flashing scales called elytra. These marine worms, often found crawling on echinoderms or inside crevices, are covered in protective scales that can be shed individually if the worm is attacked or handled roughly by a predator. Once detached, the scale doesn't just drift away, it begins to flash rhythmically and intensely, pulsating with light for a prolonged period, due to a specialized neural circuitry within the scale itself that remains active even after separation. This glowing, flashing decoy captures the predator's attention, leading it away from the worm's main body, which typically remains dark and scuttles quickly to safety in the shadows. The worm can eventually regenerate the lost scales, making this a renewable defense mechanism. Interestingly, the bioluminescence in these scales is produced by a photoprotein that is triggered by oxygen-free radicals, a unique chemical pathway that results in a very specific spectral emission. This strategy of sacrificing a part to save the whole highlights the extreme evolutionary pressures of the dark ocean, where a single flash of light can be the difference between life and death.
It is a curious paradox of nature that while the oceans are teeming with living light, the freshwater realms of our planet remain almost entirely dark, void of this biological brilliance. In the vast majority of lakes, rivers, and streams, bioluminescence is completely absent, a fact that has puzzled evolutionary biologists for decades. The prevailing theory suggests that freshwater environments are often too turbid, too muddy, or too ephemeral in geological time for the complex visual signaling systems of bioluminescence to evolve and stabilize. However, nature always seems to carve out a single stubborn exception to prove the rule, and that exception is found only in the swiftly flowing streams of New Zealand's North Island.
Here lives the freshwater limpet known as Latia narratoides, the only known freshwater creature in the entire world capable of producing light. Unlike the sophisticated flashes of fireflies or the counter-illumination of deep-sea fish, Latia defends itself by secreting a glowing, luminous slime when disturbed. This slime does not flash, but glows with a steady, eerie green light, likely intended to startle predators like eels or crayfish, giving the limpet a moment to cling tighter to its rock or escape. The chemistry it uses is also unique, involving a specific luciferin not found in any other organism, suggesting that this freshwater light evolved completely independently, a solitary beacon in the dark rivers of the world. Moving from the riverbeds to the damp terrestrial gardens of Southeast Asia, we encounter another rare anomaly in the distribution of living light, specifically within the world of mollusks. While many marine snails and cephalopods glow, land snails are notoriously dim, with one spectacular exception found in the tropical districts of Singapore and Malaysia. This is the Quantula striata, commonly known as the Diacan lantern snail, and it holds the title of being the only land snail known to flash light rather than just exude a continuous glow. What makes the Quantula particularly fascinating to researchers is that its eggs glow continuously, a trait likely serving as a warning signal of toxicity to potential predators. As the snail matures, the continuous glow fades and is replaced by the ability to pulse light from an organ near its mouth. This flashing behavior is most active when the snail is moving and feeding, leading scientists to believe it may help the snails stay together in loose aggregations, or perhaps communicate in the dense undergrowth of the rainforest. The light is a soft yellow-green hue, and seeing these small, mobile lanterns navigating the leaf litter is a surreal reminder of how diverse and unexpected terrestrial bioluminescence can be. Digging deeper into the soil, specifically in the American South, we find that the earth beneath our feet can sometimes mirror the stars above. In states like Georgia and Florida, there exists a species of earthworm known as Diplocardia longa, which can grow to impressive lengths, sometimes exceeding half a meter.
These are not your average garden worms. When these subterranean giants are disturbed or handled, they exude a copious amount of thick, sticky fluid from their coelomic cavity that glows with a brilliant bluish-green light. The intensity of this bioluminescence is startlingly high, enough to read a watch face in total darkness. The biological purpose here is almost certainly defensive. For a predator like a mole or a shrew tunneling through the dark earth, a sudden burst of bright alien light combined with a sticky secretion is a powerful deterrent. The sheer energy required to produce this light is significant, implying that the cost of the glow is worth the survival benefit. The biochemistry of Diplocardia is also distinct, utilizing a copper-containing protein that differs significantly from the iron-based systems found in other glowing organisms, further highlighting the evolutionary creativity found in the soil. Ascending from the lowlands to the higher altitudes of the Sierra Nevada mountains in California, the night reveals a different kind of glowing armor. Here, the darkness is punctuated by the spectral glow of Mottixia millipedes, often called the Sierra luminous millipedes. These multi-legged arthropods are blind, navigating their world through touch and chemistry, yet they produce a continuous, bright glow from their exoskeleton. Unlike fireflies that can control their flashes, the Mottixia glows constantly, a permanent warning sign etched in light. This is a classic example of aposematicism, or warning coloration, but adapted for the night. The millipedes produce cyanide as a chemical defense, a potent toxin that makes them foul-tasting and lethal to small predators. The light serves as a do-not-eat sign visible to nocturnal hunters like rodents, who have learned to associate the glow with the bitter taste of poison. Experiments have shown that glowing millipedes are attacked significantly less often than non-glowing decoys, proving that in the pitch-black forests of the mountains, being visible is actually safer than being hidden, provided you have the chemical weaponry to back it up.
The distribution of bioluminescence extends deeply into the fungal kingdom, creating what is historically known as foxfire, a phenomenon that has sparked folklore for centuries. There are over 70 known species of bioluminescent fungi, and their distribution tells a story of climate and genetics.
The majority of these glowing mushrooms, such as the ghost fungus of Australia or the Mycena species of Brazil, are found in the tropics, where the warm, humid nights allow for year-round growth and decomposition. However, one of the most famous species, Panellus stypticus, or the bitter oyster, reveals a curious geographical split. This fungus grows on decaying wood across the northern hemisphere, but only the strains found in eastern North America are bioluminescent. If you were to find the exact same species in a European forest, it would remain dark. Scientists have discovered that the European strains possess the genetic code for bioluminescence, but have a mutation that acts as an off switch. The American strains, however, glow with a steady intensity to attract nocturnal insects like beetles and moths, which then inadvertently pick up the mushroom spores and disperse them to new locations, ensuring the survival of the fungus in the vast dark woodlands. As you imagine these silent glowing woodlands, I'd love to hear your thoughts. If you could witness one of these rare bioluminescent events in person, would you choose the silent glowing forests of the tropics or the vast starry expanse of the milky seas in the open ocean? Let me know in the comments. Leaving the land behind and returning to the deep ocean, we find that bioluminescence is not limited to small invertebrates. It scales up to some of the ocean's apex predators. In the deep waters off the coast of New Zealand, scientists have documented the kitefin shark, which has recently been confirmed as the largest known glowing vertebrate on Earth. Reaching lengths of nearly 6 feet, this shark patrols the mesopelagic zone, the twilight layer of the ocean where sunlight fades into nothingness. The kitefin shark is equipped with thousands of tiny light-producing photophores embedded in its skin, primarily on its underside. This placement suggests a strategy known as counter-illumination. By glowing softly from below, the shark blends in with the faint light filtering down from the surface, effectively erasing its silhouette to prey looking up from below. However, the kitefin shark also has the ability to glow from its dorsal side, leading to speculation that it may use light to illuminate the dark water's floor to hunt, or perhaps to signal to other sharks in the eternal gloom. The realization that a creature as large and formidable as a shark can also be a source of living light fundamentally shifts our understanding of the deep-sea ecosystem. While the kitefin shark uses light to hide, other marine organisms use it to dazzle and confuse, and few are as beautiful as the sea pansies, or renilla, found in the sandy, shallow waters of the Americas. These are not plants, but colonies of polyps related to corals and jellyfish. A sea pansy looks like a purple, heart-shaped leaf lying on the sand, but when stimulated by the touch of a predator or the turbulence of a wave, it unleashes a spectacular display of frenzied green light. This is not a simple flash, but a coordinated wave of bioluminescence that ripples across the surface of the colony like a miniature concentric firework. The light is produced by an interplay of two molecules, a luciferin-luciferase reaction that produces blue light, and a green fluorescent protein, or GFP, that instantly absorbs that blue energy and re-emits it as a brilliant green. This energy transfer is highly efficient and serves to startle crabs or fish that might try to nibble on the soft colony. The discovery of these proteins in renilla and similar organisms has revolutionized medical science, allowing us to tag and track cells in the human body. But in the wild, it remains a simple, stunning defense mechanism on the lonely ocean floor. On a much grander scale, bioluminescence is responsible for one of the most legendary and rarely seen maritime phenomena, known as the Milky Seas. For centuries, sailors reported encountering oceans that glowed with a continuous, surreal, milky white radiance, extending as far as the eye could see, distinct from the brief sparks of disturbed plankton. For a long time, these reports were dismissed as tall tales or optical illusions. However, modern satellite imagery has confirmed that Milky Seas are very real and can cover areas of ocean larger than the state of Connecticut. This phenomenon is caused by a massive bloom of bioluminescent bacteria, particularly Vibrio harvii, living in association with a microalgae bloom. Unlike dinoflagellates that flash only when disturbed, these bacteria glow continuously through a process called quorum sensing.
When their population density reaches a critical mass, they chemically signal each other to turn on their lights simultaneously.
The result is a surreal, glowing ocean surface that can persist for days or even weeks, primarily observed in the northwest Indian Ocean. It is one of the only biological phenomena on Earth that is purely created by light and is large enough to be clearly visible from space. Traveling from the warm Indian Ocean to the freezing waters of the Antarctic, we find that bioluminescence is a crucial survival tool even in the coldest environments on Earth. The Antarctic krill, Euphosia superba, is a small crustacean that forms the bedrock of the Southern Ocean's food web, feeding whales, seals, and penguins. These krill possess highly developed photophores that can rotate and focus light using a lens system, much like a flashlight. The control they have over their bioluminescence is extraordinary. They can adjust the intensity of their glow to perfectly match the ambient light levels of the water around them. In the massive swarms that these krill form, which can stretch for kilometers and contain millions of tons of biomass, the collective bioluminescence creates a faint, shimmering aura in the dark, icy waters. This light aids in keeping the school cohesive, ensuring that individuals do not stray from the safety of the group. It is a reminder that even in the most inhospitable, freezing waters, life has found a way to use light to maintain order and connectivity. Finally, we must address the color spectrum of this global phenomenon. As we have traveled from land to sea, you may have noticed that the vast majority of marine bioluminescence is blue or blue-green. This is because blue light travels the furthest through seawater, making it the most efficient color for communication. However, in the deep ocean, there is a rare and striking exception found in the genus Tomopterus, the type of transparent planktonic worm. While almost everything else in the abyss glows blue, Tomopterus releases a cloud of glowing particles that are distinctly yellow. This yellow bioluminescence is incredibly rare in the marine world. The worm achieves this not through the standard luciferin reaction alone, but possibly through a specialized fluorescence that shifts the light toward the yellow spectrum. The evolutionary reason for this yellow light is a subject of intense curiosity.
Since many deep-sea creatures lack the visual pigments to see yellow light, it is possible that Tomopterus uses this color as a private channel of communication, a secret frequency that its predators cannot see, or conversely, to startle predators who are only adapted to seeing blue, presenting them with a color that is confusingly alien in the monochrome deep. One of the most profound realizations about bioluminescence is that it is not a singular miracle that happened once in the history of life, but rather a recurring stroke of genius that nature has invented over and over again. Evolutionary biologists call this convergent evolution, a process where unrelated organisms independently develop similar traits to solve the same problems. When we look at the genetic family tree of life on Earth, it becomes clear that the ability to produce light has evolved independently at least 40 separate times, and some estimates suggest it could be as many as 90 times across different lineages. This is a staggering frequency when you consider how complex the chemical machinery is. It means that for a jellyfish, a mushroom, a beetle, and a shark, the evolutionary path to light was completely distinct, yet they all arrived at the same radiant destination. It is as if the darkness of the natural world is a problem that life is desperate to solve, and whenever the conditions are right, biology inevitably stumbles upon the secret of illumination. This repeated invention suggests that light is one of the most useful and universally successful tools for survival in the biosphere, particularly in the ocean, where it has appeared in nearly every major phyla of animals. The origins of this light are deeply tied to the history of our planet's atmosphere, leading to a fascinating theory known as the oxygen toxicity hypothesis. This theory proposes that bioluminescence didn't actually start as a way to make light. Instead, it began as a survival mechanism to deal with poison. Hundreds of millions of years ago, as oxygen levels in the atmosphere and oceans began to rise, this reactive gas was toxic to many primitive anaerobic organisms. The chemical reaction that produces bioluminescence consumes oxygen, combining it with a molecule called luciferin. Scientists believe that early life forms may have evolved this reaction simply as a way to scrub dangerous oxygen out of their cells to prevent it from damaging their DNA.
In this ancient context, the light produced was just a waste product, a meaningless byproduct of an antioxidant defense system. It was only much later, as organisms developed complex eyes and lived in darker environments, that evolution co-opted this waste light, turning a chemical shield into a beacon for communication and hunting, transforming a survival necessity into a sophisticated visual language. As we trace these evolutionary pathways, a stark contrast emerges between the land and the sea, revealing how the environment dictates the evolution of light. In the ocean, bioluminescence is the rule rather than the exception, with over 3/4 of deep-sea animals possessing the ability to glow. On land, however, it is remarkably rare, restricted mostly to a few families of insects, like fireflies and railroad worms, and certain fungi. This disparity exists because the ocean offers a continuous, unobstructed medium where light can travel long distances without being blocked by trees, rocks, or terrain. In the vast, featureless, three-dimensional void of the deep sea, a flash of blue light is the most effective way to signal presence or find a mate across great distances. On land, however, the landscape is cluttered, and the sun dominates the visual world for half the day, making weak biological light less evolutionarily advantageous. Furthermore, the chemistry of the ocean supports the external release of glowing clouds for defense, a tactic that is physically impossible in the dry, windy air of the terrestrial world, limiting land-based glowing to internal organs and steady burns. The independence of these evolutionary events is most clearly seen in the enzymes themselves. If all glowing creatures shared a single ancestor, we would expect their light-making enzyme, luciferase, to look roughly the same, much like how the protein for hemoglobin is similar across mammals. Instead, the luciferases found in different groups of animals are radically different from one another. The luciferase in a firefly is structurally unrelated to the luciferase in a sea pansy, and neither bears any resemblance to the bacteria luciferase found in the glowing organs of a squid. They are completely different proteins, folded in different shapes, encoded by entirely different genes. This confirms that nature didn't just pass down a blueprint for a flashlight, it built a new flashlight from scratch every single time.
It is a testament to the versatility of proteins, showing that there are many different chemical ways to achieve the release of a photon. Nature is an opportunistic inventor, using whatever genetic materials are at hand in a specific lineage to craft a light-producing engine that fits the unique needs of that species. While the enzymes are unique, the fuel, the luciferin, tells a story of thriftiness and theft. Evolution often favors the path of least resistance, and for many marine animals, it turned out to be easier to steal light than to make it. There is a specific type of light-emitting molecule called Vargula luciferin, named after the tiny glowing seed shrimp or ostracods that produce it. These ostracods are abundant in the ocean, and they are a popular snack for many larger predators. Over millions of years, several different species of fish, including the singing midshipman fish, lost the ability, or never evolved the ability, to synthesize their own luciferin. Instead, they evolved to obtain it strictly through their diet. By eating the glowing seed shrimp, they charge their own biological batteries, storing the stolen chemicals in their own light organs to use later. This creates a complex ecological dependency, where the ability of a fish to glow, and therefore to mate or hide, is entirely dependent on the abundance of a tiny crustacean in the food web, weaving the evolution of predator and prey into a single glowing tapestry. For other organisms, the evolutionary shortcut was not theft, but partnership. This is the origin of symbiotic bioluminescence, a phenomenon that likely began as a parasitic infection. Eons ago, glowing bacteria may have infected the tissues of a larger marine animal. Over generations, what started as a disease evolved into a mutually beneficial arrangement. The host animal provided a safe, nutrient-rich home for the bacteria, and in return, the bacteria provided continuous light. We see the apex of this evolution in creatures like the anglerfish and the Hawaiian bobtail squid. These animals have not just tolerated the bacteria, they have evolved specialized organs, often complete with lenses, shutters, and reflectors specifically designed to house and control these microbial tenants. The squid even has a mechanism to flush out 95% of the bacteria every morning to prevent overpopulation, only to let them regrow by nightfall.
This represents a distinct evolutionary pathway where the animal's genome doesn't code for light, but rather codes for the architecture that harnesses the light of another species. Focusing on the terrestrial realm, the evolution of fireflies offers a glimpse into how a trait can shift its purpose over time. Genetic analysis suggests that bioluminescence in beetles first evolved in the larval stage, not the adult stage. The glowworms, or firefly larvae, are often bitter and toxic to predators. Their glow originally evolved as an aposematic signal, a warning label in the dark that screams, "I taste terrible, do not eat me." This defensive glowing existed for millions of years before it was ever used for romance. As evolution progressed, this warning light was retained into adulthood in some lineages. Eventually, the adults began to modify the signals, turning a steady warning glow into the complex flashing codes we see today. This shift from defense to courtship is a classic example of evolutionary acceptation, where a trait that evolved for one specific function is later hijacked and refined for a completely different use, transforming a do not touch sign into a come hither signal. However, evolution is not a one-way street toward more complexity. It is a ruthless accountant that balances cost against benefit. Just as light has evolved independently many times, it has also been lost many times. Producing light is energetically expensive. It requires the synthesis of complex molecules and the burning of precious calories. If a species moves into an environment where the light is no longer necessary, perhaps they migrate to shallower waters where sunlight is abundant, or they evolve a lifestyle that relies less on visual signaling, natural selection will quickly eliminate the trait to save energy. We can see the scars of this loss in the genomes of certain ponyfish and shark species, which have close relatives that glow but have themselves gone dark. Their DNA often contains the broken remnants of luciferase genes, fossil genes that no longer function, serving as a silent record of a time when their ancestors once illuminated the dark, proving that in the economy of nature, if a light doesn't pay its way, it is extinguished. One of the strongest evolutionary pressures driving the development of these complex light organs is the need for camouflage, specifically the counter-illumination strategy we touched upon earlier. This pressure has shaped the very anatomy of countless deep-sea species. In the mesopelagic zone, the main threat comes from below, from predators looking up against the faint twilight of the surface. This specific danger forced the independent evolution of ventral photophores, light organs concentrated solely on the belly in unrelated groups like lanternfish, hatchetfish, and cookie-cutter sharks. It is a remarkable example of convergent evolution driven by the physics of light and shadow. The need to erase a silhouette forced these different animals to develop the same solution, a tummy that glows. It even influenced their internal structures. Many of these fish have evolved reflective linings inside their bodies to ensure every photon is directed downward, wasting nothing, sculpting their anatomy around the strict requirements of invisible survival. Finally, when we try to pinpoint exactly when this phenomenon first appeared, we must look into the deepest reaches of geological time. Because soft-bodied organisms do not fossilize well, the physical record is sparse, but molecular clocks allow geneticists to peer back hundreds of millions of years. Current evidence suggests that bioluminescence is an incredibly ancient trait, potentially appearing as early as the Cambrian period or even before. Some of the oldest glowing lineages are likely the ctenophores or comb jellies. These gelatinous, drifting creatures are among the most primitive of all animals, branching off near the very base of the animal family tree. The fact that they possess their own unique form of light-producing proteins suggests that bioluminescence was one of the earliest complex functional traits to arise in the animal kingdom, illuminating the primeval oceans long before the first fish swam or the first plant took root on land. Thinking about these ancient oceans, filled with silent, drifting light, is a reminder of how deep and strange the history of our planet truly is. As you lie there in the quiet dark, listening to the story of the Earth, do you find the idea of the deep, dark ocean comforting and cozy? Or does the thought of such vast, silent emptiness feel more mysterious to you? The quest to harness the living light of nature has moved from the realm of science fiction into the tangible soil of our gardens, beginning with the dream of creating self-illuminating plants.
For years, scientists have attempted to create glowing flora, initially by borrowing genes from fireflies, but these early attempts were faint and required the plants to be fed special chemicals to shine. However, a major breakthrough occurred when researchers looked instead to the kingdom of fungi, specifically a bioluminescent mushroom called Neomythopanis nambi. By identifying the metabolic pathway these mushrooms use to convert caffeic acid, a compound already present in all plants, into luciferin, scientists were able to engineer tobacco plants that glow entirely on their own, without the need for external chemical food. This creates a closed loop where the plant converts its natural building blocks into light and then recycles the byproducts back into fuel, glowing continuously with a visible green aura that can be seen by the naked eye. This technology brings us closer to a future where our pathways might be lined not with electric bulbs, but with tall, gently luminescent trees that guide our way through the dark. Beyond the aesthetic beauty of glowing gardens, the medical applications of bioluminescence are revolutionizing how we detect and treat diseases, particularly cancer. In the laboratory, researchers have developed techniques to tag cancer cells with the luciferase enzyme, effectively making the disease light up in the darkness of the body. When these tagged cells are introduced into a model organism, sensitive cameras can detect the light emitted from within the tissue, allowing scientists to track the growth of tumors and the spread of metastasis in real time without invasive surgery. This method is far more sensitive than traditional imaging in the early stages, as even a small cluster of glowing cells can be detected against the dark background of healthy tissue. It allows for the testing of new drugs with unprecedented precision. If the light dims, the drug is working and the cancer is dying. This use of living light serves as a beacon of hope, illuminating the hidden behaviors of disease and guiding the development of therapies that could save countless lives. While we look inward at the human body, bioluminescence is also being deployed to watch over the health of our external environment, serving as a microscopic alarm system for water quality.
Certain species of bioluminescent bacteria, such as Vibrio fischeri, are naturally sensitive to toxins. Their glow is directly tied to their metabolic health, meaning that when they are happy and thriving, they shine brightly, but when they encounter pollutants, their light instantly dims. Scientists have harnessed this trait to create rapid toxicity sensors for testing drinking water and industrial wastewater. Unlike standard chemical tests that look for specific known contaminants, these bacterial sensors provide a broad-spectrum warning. If there is anything in the water that harms living things, the light goes out. It is a poetic application of biology, where the fading of a tiny bacterial light warns us of unseen dangers, acting like the canary in the coal mine, but for the microscopic world of aquatic chemistry, ensuring the safety of the water we rely on. This fascination with biological light has also sparked ambitious architectural visions, leading to research into replacing streetlights with bioluminescent infrastructure to reduce global energy consumption and light pollution. The concept, often referred to as planetary energy, envisions a world where the harsh, artificial glare of LEDs is replaced by the soft, ambient glow of genetically modified biological systems. Researchers at MIT have experimented with infusing watercress plants with specialized nanoparticles that carry luciferase and luciferin, turning the leaves themselves into passive light sources. While currently these plants only glow for a few hours and emit a dim light, the theoretical physics suggests that optimizing the packaging of these enzymes could eventually produce enough light to read a book by. Imagine walking through a city at night where the light doesn't rain down from electric poles, but radiates softly from the lining of the streets and the leaves of the trees, a vision that would dramatically lower our carbon footprint and restore the natural darkness of the night sky for migrating birds and stargazers alike. As we imagine this glowing future, it leads us to consider the mysteries of our own minds, where bioluminescence is currently helping to map the most complex structure in the known universe, the brain. Neuroscientists use a tool called GCamp, which is a genetic fusion of a green fluorescent protein and a calcium-binding protein, to watch neurons fire in real time. When a neuron activates, calcium floods into the cell, causing the protein to change shape and flash brightly, effectively converting electrical thought signals into visible flashes of light. This allows researchers to watch thoughts and memories ripple across the brain of a living organism, creating a literal map of neural activity. It is a stunning convergence of nature's tools, using the protein from a jellyfish to visualize the spark of consciousness. I'd love to know, if you could have any object in your room glow naturally like these organisms, providing a soft, living nightlight while you sleep, what would you choose? Maybe a glowing plant in the corner, or a ceiling painted with living stars? Let me know in the comments. The utility of these light-emitting enzymes extends into our daily safety, specifically in the food and hygiene industries, through a technology known as ATP bioluminescence testing. Adenosine triphosphate, or ATP, is the energy currency of all living cells, from bacteria to human skin. Sanitation experts use portable swabs containing firefly luciferase to test surfaces in hospitals and restaurant kitchens. When the swab picks up even invisible traces of biological residue, the ATP reacts with the luciferase to produce light. The amount of light generated is directly proportional to the amount of contamination present, providing an immediate pass-or-fail result for cleanliness. This means that the same chemical reaction that allows a firefly to signal a mate in a meadow is used every day to ensure that surgical tools are sterile and that food processing equipment is free of harmful bacteria, a silent guardian of public health rooted in the chemistry of an insect. Diving deeper into the molecular toolbox, scientists utilize a phenomenon called bioluminescence resonance energy transfer, or BRET, as a molecular ruler to measure distances between proteins within a single cell. In this sophisticated technique, a bioluminescent molecule is attached to one protein and a fluorescent acceptor molecule is attached to another. If the two proteins interact or come extremely close to one another, the energy from the bioluminescent donor is transferred non-radiatively to the acceptor, causing it to emit light of a different color. This color change signals that an interaction has occurred, allowing researchers to study how drugs affect cellular receptors or how viruses bind to host cells with nanometer precision.
Unlike other methods that require firing lasers at the sample, BRET generates its own light, which avoids damaging sensitive tissues and provides a purely biological way to spy on the secret handshakes occurring between the molecules of life. However, the expansion of bioluminescence into the wild brings with it ethical and ecological questions regarding genetic pollution and the disruption of natural night cycles. There is a valid concern among ecologists that releasing genetically modified glowing plants or animals into the wild could wreak havoc on existing ecosystems, particularly for nocturnal insects that rely on darkness and specific light cues for navigation and mating. If a modified glowing weed were to escape a garden and proliferate in a forest, it could act as a perpetual light trap, confusing moths and disrupting the predator-prey balance that has evolved over millions of years. This potential impact forces us to tread carefully, balancing our desire for aesthetic and functional biological light with the need to preserve the sanctity of the natural night, ensuring that our attempts to beautify the world do not inadvertently blind the creatures that already live in the dark. Despite these challenges, the cultural and artistic integration of bioluminescence continues to grow, merging biology with fashion and design in a movement often called bioart. Designers are experimenting with fabrics woven with bacterial colonies that glow when fed specific nutrients, creating garments that are literally alive and illuminate the wearer with a ghostly organic radiance. There are installations where glass vessels filled with bioluminescent algae respond to the vibration of human voices or the touch of a hand, creating a reactive light show that bridges the gap between the viewer and the natural world. This artistic exploration serves a dual purpose. It creates a sense of wonder that technology alone cannot achieve, and it familiarizes the public with biotechnology, transforming the cold image of a laboratory into something warm, intimate, and profoundly human, suggesting a future where our technology is grown rather than built. Ultimately, the 100th and final fact about bioluminescence is that it represents one of the most profound examples of convergent evolution, a testament to life's relentless drive to dispel the darkness.
It has evolved independently at least 94 separate times across the tree of life, appearing in bacteria, fungi, fish, squids, and insects, each time solving the problem of darkness with a unique chemical invention. This universal recurrence suggests that light is not just a physical phenomenon, but a biological imperative. Life, in its infinite adaptability, finds a way to shine. From the crushing pressure of the deep sea to the silence of a forest floor, and now to the forefront of human medicine and technology, bioluminescence reminds us that even in the deepest shadows, there is the potential for brilliance. It connects us to the cosmos, mirroring the stars above with the sparks below. And as you drift off to sleep, you can rest in the knowledge that the world is filled with living light, watching over the night. Thank you for joining me on this illuminating journey through the wonders of bioluminescence. If you enjoyed relaxing with these facts and want to hear more calming stories from the natural world to help you sleep, please subscribe to the channel so you never miss a video. Good night.